Well, welcome back, folks. We are going to continue our work over DNA today by talking a little bit about the process in which DNA makes a copy of itself. This is known as DNA replication, and this is absolutely crucial for your survival. So think about it. Your cells have to make copies of themselves. You're growing young boys and girls. You're replacing old cells that have died. So the DNA inside of those new cells has to come from somewhere, and that's when your DNA makes a copy of itself. It's also important because we start off with 46 chromosomes, and if we were to split those in half each time your cell would undergo replication or mitosis, then you'd have half the number of chromosomes each time it did that. So you'd go from 46 to 23 to 11 and a half to 5.75 and so on. So it's really important that DNA has the ability to make a copy of itself. And we're going to talk about that process today in this video. Hopefully by the end of this video, you should be able to understand why DNA needs to replicate, why DNA needs to make a copy of itself. And you should also be able to explain the process of DNA replication, that is the individual steps that occur in order for DNA to replicate and make a copy of itself. So let's take a look at our vocabulary for this particular video. We're going to be focusing on DNA replication. We'll talk about DNA helicase, the replication fork, something known as a single-stranded binding protein, DNA polymerase, DNA ligase, 3' and 5' ends, and my favorite vocab term, Okazaki fragments. So why does DNA have to replicate? Well, cells replicate. Cells make copies of themselves all the time. And so DNA has to be able to do the same. Now, how this occurs is what's known as semi-conservative replication, which means that each original strand acts as a template to build the opposite strand to produce identical DNA molecules. So what you notice is that the DNA breaks apart. Essentially, the double helix structure, you got two strands, those hydrogen bonds that hold the strands together break apart. And then each individual strand serves as a template to make a new one. And so what happens is that DNA replication occurs on both strands of the original DNA at the same time, eventually producing two different DNA molecules. Now, the place where this splits is known as the replication fork. And in eukaryotes, there are hundreds, even thousands of replication forks that occur at a time because eukaryotic organisms have more DNA. Prokaryotic cells, however, only have one replication fork. Their DNA is just in a circle, so it's very easy for one replication fork to make a complete copy of their DNA. But with eukaryotes, because they have so much more DNA, they have to have multiple replication forks for this to occur. So what's the first step? Well, DNA helicase, hopefully you notice it ends in ase, which tells you that it is a protein, more specifically an enzyme. DNA helicase unwinds and unzips the DNA by breaking the hydrogen bonds, thus creating the replication fork. So the DNA helicase almost acts like a zipper. It unzips the DNA, moving in a direction. Now here in this picture, the DNA helicase is going to move to the left. Replication will occur once the DNA has separated from its opposite strand. SSBPs, which stands for single-stranded binding proteins, they prevent the DNA from rewinding. So you think DNA wants to be in that double helix shape. And so by unwinding it, it really wants to push it back together. But single-stranded binding proteins attach to the outside ends of the strand and chemically alters the DNA, which prevents it from wanting to join back together again. So how does this work? On the leading strand that runs from 3 to 5' as you're going from left to right on your screen, that is made continuously. So DNA polymerase adds complementary nucleotides. So polymerase is the one that recognizes each part of the DNA and says, oh, there's an A, we need to add a T nucleotide here. Oh, there's a G, we need to add a C nucleotide here, and so on going down the line. The leading strand is replicated continuously as the polymerase follows the helicase. So in this picture, as the helicase moves to the right, the polymerase moves along with it on the top strand, just following it along, making that complementary strand of DNA. So the 5' to 3' end is made continuously. The problem with this is when we get to our lagging strand, because DNA can only be made in the 5' to 3' direction. Well, that becomes a problem when we look at our lagging strand. On our lagging strand, DNA polymerase moves the opposite direction. Again, DNA can only be made from the 5' to the 3' direction. So the polymerase actually moves the opposite direction on the lagging strand. So it moves away from helicase. Well, as helicase exposes more DNA, that polymerase is not going to replicate it. So what happens is it creates small pieces of DNA called Okazaki fragments. So a small piece of DNA is made moving to the left, and as more DNA to the right gets exposed, the polymerase will detach, go back to the replication fork, and then move the opposite direction. So it kind of makes it in a bunch of little fragments. So it makes a little fragment, jumps back to the replication fork, makes a little fragment, jumps back to the replication fork, and so on. Now these DNA molecules are bound together using DNA ligase. A ligand is kind of like a biological glue. So ligase is an enzyme that helps bond the Okazaki fragments together. So this is really important to understand, is that the leading strand is made continuously because it just follows the helicase. It makes DNA from the 5' to 3' end. The lagging strand makes DNA from the 5' to 3' end as well, but unfortunately, because DNA is anti-parallel, it goes the opposite direction of the replication fork. So it makes small individual segments known as Okazaki fragments. It'll make a little tiny fragment and then jump back, make a little tiny fragment and then jump back. And this process repeats itself until the entire strand of DNA is replicated. So again, just kind of keep in mind that the replication fork moves to the left here as the helicase unwinds the DNA. If the polymerase moves from 5' to 3', the top strand will be made continuously, while the bottom strand will be the lagging strand, is made in small fragments called Okazaki fragments. Polymerase adds the A's, T's, C's, and G's, while ligase binds the Okazaki fragments to one another to make one continuing piece of DNA on the lagging strand. So it's a bit complicated, but we'll talk more about leading and lagging strand as we come back as a class. Keep in mind that DNA must replicate because cells do. As cells make copies of themselves, we need to make more DNA in order to have DNA inside of those cells to be able to function. DNA replication is semi-conservative, meaning that each original strand acts as a template to build a new strand. A variety of enzymes play a role in this process, and each strand is made differently as the polymerase moves opposite directions on each strand. And the leading strand, it follows the helicase, and on the lagging strand, it moves opposite the helicase. So it has to be made in small fragments called Okazaki fragments. And each new DNA molecule that is created is genetically identical to the original one. And that's really important. We want to make sure that all of the DNA inside of our cells is exactly the same. If there's not, or there are problems with this, that can lead to some very significant health problems for the cell and the organism. So hopefully you have a little bit better understanding of DNA replication. We'll talk a little bit more about this in class and move on to protein synthesis next class period. We'll talk to you later, guys. Bye-bye.